a lot of people still don't know very much about them. And then a little bit about pattern recognition at the end. But I just want to highlight that Alistair Curd, who developed that software, is going to talk about it in a couple of weeks' time on the 1st of November. So he's really going to talk about it in some detail and do some live demos, he tells me, which all sounds terribly exciting. So I thought I'd leave most of that to him. So thank you very much for the introduction. Actually, it's quite interesting because there's sort of picture pop up of a person who's actually here. But I just thought I'd thank the people first, because I'm going to talk about stuff that we've been doing over the last few years, and there's quite a lot of different people who've been and gone from my lab involved in the imaging. So Ruth, who was a PhD student and then briefly a fellow, who she now works in our bioimaging facility. Anna, who was a PhD student at Wellcome Trust, NIH student, who's now doing a postdoc in Germany. Alexa, who was briefly a Wellcome Trust funded ISSF fellow in the lab, and is now doing some sort of scientific writing. And Fran, who's still in the lab and actually working a lot on Aphromoos at the moment. Alistair, who you'll hear about in a few weeks' time, who's the physicist who builds our microscopes and does the analysis. Oh, that's got the wrong name. The names have been transposed. This is actually Brendan. He was a PhD student that did some of the work on Aphromoos. Glenn is also helping to work with Aphromoos. Joanna helps to do the software, and she has helped to do some crystal structures. Those are some collaborators that I'll just very briefly share a little bit of work on cilia. And these are the guys who do the Affimer screening. So if you're really interested in Aphromoos and you want to use them, Darren is the person to approach, who I can put you in contact with. Christian, who also really runs the facility, and Anna, who works in the facility doing screening for us. My super resolution journey started when I went to do a bit of a sabbatical at NIH and met Harry Schroff, who's been very supportive and has helped us develop our super resolution. Similarly, Suliana and Jonas and Ralph and Thomas, who was a PhD student in Ralph's lab, all helped us on with the super resolution. So many thanks to those, and of course to our funders. So as we started doing super resolution imaging, actually we started with Palm and using EOS. But of course, a lot of what you want to do is with antibodies, because that's how we all image cells. We get our antibodies out the freezer, and then we label up our favorite protein of interest, and then we go to the microscope and image it. But the problem with super resolution imaging is that antibodies don't work as well. And the main reason for that is because of their size, because a primary antibody and a secondary antibody on the top means that you put your dye, which is what you're actually imaging, a long way away from your actual target. The large size of those antibodies means they can be excluded. So if you're looking at a dense region of the cytoskeleton, they might not get in, so they might not even label, or they might only label the outside and not the inside of what you're looking at. And there is quite a lot of uncertainty about where the dye label is. So which I'll come on to and talk about, or even how many dye labels you've got. So if you want to X marks the spot, that's really not very good, because X doesn't really mark the spot. And this is a nice paper that just came out where it was really aimed at looking at improving the labeling of antibodies using this glutamine modification, because typically when you label antibodies you use lysines. There are a couple of other methods that you can use, but most common way is to use lysine. So this was a different way of modifying the antibodies, which is deglycosylating them and labeling on the glutamines. Then these were tested in DStorm and DNA Paint. And what they actually ended up finding is that quite a good estimate of what the linkage error is, but also the fact that these antibodies have quite a big radius, so that the dye molecule can sort of flop about because of this flexible hinge between the different domains of the antibody. So this list there, which I've whipped out of their table, is the same order as here. So you can do an NHS linkage, you can look at secondary and primary, or primary only, and this is the equivalent but for DNA Paint. And the linkage errors can get quite large, as you can see, so up to about 30 nanometers. Now, if you're imaging a structure that's only got a radius of 12.5 nanometers, you really are putting your dye a long way away from the molecule. So I think this is a problem. So the question is, what can we use instead? And if we want to get away from using primary and secondary antibodies, well, yes, we could just directly label the primary antibody, but it's still quite big. People have started to shift to nanobodies. But what we do in Leeds is we use these things called Affimers, and that's really because Affimers were developed in Leeds, and they have some advantages over trying to make an antibody. Nanobodies are not exactly trivial to make because you have to get hold of your shark or your llama or some naive library, and then extract the RNA, clone it into some sort of phage display library, screen it, eventually find some antibodies that work against your target protein, and then you can express it. But it is an antibody, it does still have cysteines in it, even though people are modifying that and trying to make some cysteine antibodies. But the key thing is that it's much smaller, because some of the antibodies that you get in the sharks and the cavalids are just the heavy chains, so no light chains, and they'll also work as a single heavy chain, and that's a single heavy chain that makes the nanobody. So Affimers are not antibodies at all. Antibodies are an artificial protein, and they were made in Leeds originally by Mike McPherson and Darren Tomlinson. They are based on a plant protein, and what they did was take all the sequences from all the plant proteins and generate an optimized sequence, and then express that, and it's engineered so it's really thermally stable. So in fact, in purifying it, one of the steps is to heat the bacteria lysate up, and it denatures a lot of the proteins, but the Affimers come through unscathed. So they created a phage library, and that has got 3 × 10⁻10 different Affimers in it. The Affimers have these 2 variable loops. They typically pull out Affimers that have nanomolar binding affinity, but the key thing is that you can express them and purify them from bacteria really, really easily. Once you find your Affimer, you can subclone it into a standard bacterial expression vector with a his tag, and you can do the standard nickel NTA affinity purification after heating up your bacterium lysate. And the other interesting thing about it is that the sequence was engineered so it doesn't have any cysteine residues in it at all, but you can add them in as you want. So if you really want to label absolutely precisely, you know where your dye level is, you just put it on the end of the C terminus, and then you can use maleimide linkage. So that means it's really easy to generate, or you can biotinylate that cysteine. So it's really easy to generate, label the Affimers. If you really want, you can still label on the lysines, but we typically put the cysteine in. So we take the library, we put our protein of interest in, we wash off the non-binders, we elute the binders, we amplify the phage. Well, the screening facility does this for us, let's be honest. And then we repan several times to get, it usually varies. So really, we typically get quite a few Affimers. When we do this ELISA, we pick some, so we usually pick no more than about 10 to actually analyse, because they take quite a long time to express, purify and analyse. But the nice thing is it's not too bad, because you can express them in 50 ml cultures, so they're really, you're not making large cultures to express and purify each of your Affimers. You can get a lot of protein from just a 50 ml culture, and they're really very stable. You can put them in the fridge. The other day I got a Tubulin Affimer out, which I'll talk about in a minute, that I made, or was made in the lab about 3 years ago, and I thought, I'll just try that Tubulin Affimer, got it out and it worked just fine. So, and it's quick, it's 4 weeks. So you give the protein of interest to the facility and you get your Affimers back in about 4 weeks. The lengthy part is actually testing them all out and checking which ones work for your particular application. So this is an example. These are Affimers that we got for actin. So we did the screening. This is the standard ELISA you get back as the final step. And you can see various different binders here in the cyan colour. So those get sent away for screening, and occasionally some of these are the same, so we take the unique ones. We took 4 of those to test. We did a pull down assay with filamentous actin, so you've got soup, which shouldn't have any filamentous actin in, and pellet, which should. Does the Affimer bind? And when you increase the concentration of Affimer, is there an increase in binding? So you can do a standard binding plot and work out what the binding affinity is. And typically, I think all of those had sub-nanomolar binding affinity, apart from this one, number 2. But 6, 14, and 24 were all pretty good. So then it's the long journey of testing. So typically in our lab, we tend to use paraformaldehyde fixation. And when we tried it out in paraformaldehyde, here is phalloidin, which works really nicely in paraformaldehyde, and here is the Affimer, and you can see the only one that worked was Affimer 14. But when we switched to methanol fixation, then they all work. So that's the first thing that we always test now, is which fixation is best for our Affimer. Affimer 2, now you remember that weak binding Affimer actually worked really well. It was really nice staining when we did the methanol fixation. So there's the antibody to actin, and here's the Affimer, and you can see that they co-superimpose pretty nicely. So even though it's a weak binder, it actually still seems to recognize actin in the contacts of methanol fixed cells. So one thing we wondered about that, and subsequently I think the screening facility has managed to get Affimers that are sensitive to the conformation of your protein, something that would be really hard to do with a sort of standard antibody approach. So we think possibly what's going on here is that the Affimers are sensitive to the F-actin conformation. And we know that there must be some sort of change in F-actin conformation because phalloidin does not work on methanol fixed cells. So there must be some change to the structure that the Affimer is recognizing in methanol that phalloidin doesn't recognize. So, and if it works, you know, we try it on lots of different cells, and this is one of the other Affimers, but this is back to phalloidin, paraformaldehyde fixation, and the fixation works for Affimer 14, and it works on every single cell type we try, whether it's human, insect, or amoeba. So if you get a good Affimer, it tends to work really, really well. So the other thing we thought we'd try for fun would be to put GFP on the Affimer and see if that would work. Would it actually label up the actin in a live cell? And this is just comparing, on one side you've got live act, and then on the other side you've got one of the Affimers, GFP Affimer 6. And it's labeling up something that looks like actin, right? So they do seem to label actin in live cells, but there is a key difference. So when we tried that Affimer with a different M-cherry actin as a co-label, and then you stimulate the cells to make them make these lovely protrusions that you can see, what you can see is the GFP Affimer doesn't actually go into that protrusion very nicely. So again, this might be consistent with some sort of conformation. So the new actin, the fresh actin, it might be typically newly polymerizing actin tends not to have tropomyosin on it, so that could be one thing. It could be a change in conformation of the actin, something. But anyway, it looks as though the Affimers are labeling up what we might call old actin rather than fresh, young, new actin. So you have to be really careful with these things and really sort of test them out. As soon as you label actin in cells, something happens. Even life act is not the perfect one because it makes lots of phylopodia, and there's some nice work from Stefan Branz's group that shows that it partially blocks myosin binding. So you have to be really careful when you label these things in cells. So anyway, so we then went on to do super resolution, and we used an Alexa 647 Affimer 14 in D-Storm. We got some nice data. You can kind of zoom in and convince yourself you can see some nicely labelled actin filaments. And there again, you can zoom in, you can see some nicely labelled actin filaments. So it seems to work very well. This is in D-Storm, of course. And we just happened to have a conversation with Ralph Jungmann about the potential for DNA paint. So they took our Affimer away. and they worked out a nice way of labelling it on the cysteine, and they tried to see if they could get it to work in DNA paint. And I think it was Thomas who mostly did these experiments. I'm not going to explain DNA paint because I'm assuming most people know what that is. And this is some of the preliminary results that they sent us. So this isn't this was before we actually got to our publication, but you can see, I mean, the preliminary results really show some very nice labelling in DNA paint. This is what made it into the final paper, and you can see very nice actin labelling, and you can see very nice labelling, actin labelling in Z as well. Okay, so the actin Affimer seems to work quite well. Of course, you can't be a super resolution microscopist and not image microtubules. So we went for a Tubulin Affimer. Now this turned out to be a lot more difficult, and in retrospect we know what we need to improve to get Tubulin Affimers. The main problem is, unknown to us, the screening facility was taking our lovely polymerized tubulin, which looked beautiful when we gave it to them, and putting it into PBS. And PBS is not good for microtubules in vitro screening. Despite that, we managed to get one really nice Tubulin Affimer, and here it is. So you can see the staining for the Affimer, the staining for tubulin. It labels up the mitotic spindle, although it seems to label up a subset of tubules, microtubules in the mitotic spindle, and you can see it in late mitosis here, also still labeling up mitotic spindle. So we had 10 Affimers in this case, and we had to screen all of them to see which one worked. And we were really getting to the point where we thought we'd never find a Tubulin Affimer, but in the end we did. This just demonstrates why the small size of the Affimer is good, because this is a cytokinetic furrow. So here's one cell, here's the other cell. Here's the Affimer staining, which goes through that furrow. And here's the tubulin antibody. So this is the Affimer to tubulin, this is the antibody to tubulin. And typically when you stain with an antibody, you always get what's called this cut zone for tubulin. And the Affimer gets inside that cut zone. And the reason why the antibody finds it hard to get in is in that cut zone, there are lots of microtubules highly bundled together. And you can see in these two EM images that it really is a dense region of cytoplasm. So we think the Affimer can get in where antibodies cannot. And then again, we tried this in DStorm. It was all done very quickly for this paper, so they're not our best images ever, but it does show you that it works, and it shows you that compared to an antibody, the profile across the microtubule is narrower, so we're reducing our linkage error. So Ralph has also had a go with the tubulin antibody. This didn't make it into the paper, but I'm just going to show this preliminary data anyway, because it does demonstrate the linkage error. So this is DNA paint with dual antibodies. This is DNA paint with a single antibody, and this is using the Tubulin Affimer. Now, Tubulin Affimer, they found it a bit harder to get it to work in DNA paint for whatever reason, but they could get at least the 2D DNA paint to work. And they did measure the diameter of the microtubules, and when you see the diameter of the microtubules, you can see that linkage error is much smaller for the Affimer than it is for either primary or dual antibody staining. So that really does tell you that you should be using smaller either nanobodies or Affimers if you're really serious about finding where your proteins are in cells using superres. So this is just the one a slide that demonstrates that you can use Affimers in any kind of imaging you want. So these are Affimers that we've made to a protein called CEP290. CEP290 is a protein that sits in the transition zone of primary cilia. It's some sort of long coil, and we don't really understand exactly what its structure is or how it assembles into the transition zone. But we have Affimers that label either end of it, so either the N terminal or the C terminal region. In Convocal, you can already start to see that there is a difference in the labeling. This is ARL13B, which is just a marker for primary cilia, and that's a merged image, but you can see there is some difference So the Aphromoos are getting into this transition zone, which is a narrow region of the cell again, so they're doing their job very nicely. This is a stud image using a Studicon that we have at Leeds, again showing some difference in staining. And then finally we can start using those Aphromoos in storms. So you can use them in any kind of imaging you like. It's not that they're just for super-res, you can use them for standard imaging. So I'm just going to summarise that briefly then. Aphromoos have some significant advantages. They're small, they're easy to generate, 4 weeks after submitting your protein of interest. You can express them in bacteria. You can put in a unique site for labelling. Because they're small, you get a reduced linkage error. Because they're small, they get into dense regions of the cell. You can use them in live cells. It doesn't always work. We tried it with the GFP as a GFP tubulin Affimer, and that didn't work. So it doesn't always work. But you can use them in all sorts of fluorescent imaging. What you also find is that different Aphromoos have different properties, so that one might label in this situation, one might label in that situation. Did I not show? You didn't see a slide of Tubulin Affimer 7, did you? Anyway, we have a second Tubulin Affimer, which maybe is in the wrong order, but that only labels up the mitotic spindle. It doesn't label interface cells. So it's a bit like getting monoclonal antibodies. When you get a lot of Aphromoos to the same protein or the same protein domain, you find that you get a diversity. So some will work in some situations, some will work in other situations. And they can be conformation sensitive Aphromoos, and they can be post-translational modifications sensitive Aphromoos as well. So if you're really interested in getting something that recognises PTMs or conformations, that's a real possibility. So the main way that we're trying to use Aphromoos at the moment is to look at the Z disk. And I think I've skipped a few slides by mistake, so there we go. So this is a cardiomyocyte, and these stripes here are Z discs. The Z discs are for individual myofibrils that align up, so each of these stripes is actually lots of sarcomeres aligned across the cardiomyocyte, and the Z discs are all lined up by connecting proteins. But these Z discs are really narrow. They're about 100 to 150 nanometers wide, so there's absolutely no way that you can really see any detail other than the line because they're below the resolution limit. So of course everybody says, why don't you just use EM? But it turns out they're tricky to image even by EM. So the latest paper that's really looked at muscle sarcomeres with some very nice work from Stefan Raunzer's group using cryo-EM tomography of fib-milled myofibrils. And even when they look at the Z disc, really the only protein they can identify is one of these cross-linked proteins called alpha-actinin. But there are 30 different proteins in the Z disc at least. So where are they all? How are they organised? And how do you find them? EM at the moment isn't working to do that. Of course, I'm sure correlated plan is being developed, but at the moment it doesn't seem to do it. So the basics of a Z disc is that you have a thin filament which has got actin in that comes in from one sarcomere over here and a thin filament from this sarcomere over here, and they have opposite polarity and they're cross-linked by alpha-actinin. And it's not alpha-actinin is not there like always there like a step ladder. It goes in and out of the sarcomere. All of the proteins are exchange in and out of the Z discs in the sarcomere, but alpha-actinin does cross-link. And then all of the other proteins, well, I've put some of them here. This is my fictitious Z disc with some sort of suggestion about how they might be organised. And you can see there are lots of question marks because there are lots of proteins in there and we really don't know how they're organised. And this is important because the Z disc is a key signalling centre for muscle. So if you go to the gym and build your muscle up, that's because you're stressing your muscles and you're putting tension on the Z discs, and all the signalling proteins in the Z discs then go into the nucleus and say, we need some more muscle, please. Make some more myofibrils or make some more protein. So understanding how proteins are organised in the Z disc is key for understanding how they work and how all the proteins interact and talk to each other. So we've been making lots of Aphromoos. At the moment, a lot of these are still at their final stages of screening, but we have Aphromoos to alpha-actinin, capping protein, myopalladin, ZASP. This is a little picture of ZASP here on some human cells that have been sectioned, etc., etc. So, and the one that we've done the most work on is the one to alpha-actinin, which is the first one we made. And this is another advantage of Aphromoos. They're quite good at crystallizing. So if you've got a protein that you want to crystallize, the Affimer can actually help stabilize that protein and help it crystallize. It turns out in this particular example, we already had CH domains that we had crystallized for alpha-actinin. So we just did the co-crystal to see where these loops bind. So also now we know exactly where the loops bind, and this is the full length alpha-actinin, which is an anti-parallel dimer, and we can see where the Aphromoos is binding, and it's away from the actin binding interface. So these CH domains are actin binding interfaces. So again, we had a lot of different Aphromoos. We tested them all. This is one of them, just in a standard confocal, and here is the 3D storm image. And just a little note of direction. So what we call X is actually going from one end of the cardiomyocyte right down to the other end of the cardiomyocyte, and Y is going along the length of the Z discs that are joined up. So this is what we call the X direction, going from one side of a sarcomere to from one sarcomere to the next sarcomere across the Z disc. So we get some sort of pattern. If we just look at a single image, it's not very informative, but of course we can use super resolution to work out the XYZ positions of all the molecules that we localize in a 3D storm image. So can we actually find out some kind of information from that? You can project it, and you can see that there looks as though there's some sort of patterning going on, there's some sort of organization. But this is where we started to, or I should say Alistair started to develop a way of getting patterns out of super resolution images. And there is Jonas's picture, because Jonas very kindly collaborated with us to see if we could do this for nuclear pores, because nuclear pores are becoming a sort of standard go-to. Can we do it with nuclear pores? Yes, the method's working, so we can use it for whatever we want to do. So it works in a very, as I say, Alistair's going to come along and give you all the technical details in a few weeks' time, but it works very simplistically. It works by when you localize each of your positions in a storm image as XYZ positions, and then you can graph those. You can look at the images of all of those XYZ positions. That's your field of analysis. That's just looking in XY. You can zoom in and see individual nuclear pores in this case, or whatever it is that you're looking at. But then what you do is you basically plot the distances between XYZ positions. So it really is XYZ. So you actually measure the distances between every point and every other point around it. You can do it across the whole image, and you get a bunch of new XYZ, and a bunch of distances, which we call relative position density. And then you can plot those in any way you like. So you can just plot the X direction, you can plot XY, you can plot XYZ, you can plot Y, whatever you like. And this is just really the relative number of for here it's XY separation. And then you start to see some peaks, and here are two key peaks at specific distances that come out. So then you have to interpret that plot, but of course for nuclear pores it's fairly straightforward, because the rough dimensions of nuclear pores is known, and we know they have rotational symmetry, and we know this particular protein we were looking at, there were 3 in each of the positions around the ring. So we could build models for different fold symmetries, 5 up to 11-fold or symmetry, and these plots you can see here are the model. So it's exactly the same. You create a load of distances, you work out the relative position densities, and then you have some different plots for the different models here, which seem to fit better than others. And you can use a statistical approach called a Keiki information criterion, which compares your model to the real data. And that's a nice spatial way of looking at comparing two distributions, and it gives you an error readout, and it tells you of the 5 models you tested which one is most likely to be true. It doesn't tell you the actual real truth, but it tells you the most likely, or out of the models you've given it, which one's most likely. So we did this for the Z-disk. So here again is the picture I showed you earlier, and this is plotting the raw data. Now we don't have quite so many localizations as we did for the nuclear pore, but you can start to see some peaks appearing. So we could create a model based on what we think is happening in the Z-disk. In the Z-disk, we think there are these alpha-actinin structures, and we think they're roughly about 19 nanometers apart as you go from one side of the sarcomere across through the Z-disk to the next sarcomere. And we think we have a second dimension here, which is the distance between the 2 matching filaments, which so they're both pointing in the same direction from the same sarcomere. And we have a third distance here, which is because alpha-actin is an antiparallel dimer, you can have a label either side of the Z-disk, the actin filament. That's roughly about 11 nanometer distance. So 24 nanometers between filaments of the same polarity from the same sarcomere, and 11 nanometers across the thin filament. And we get this main pattern coming out, which is about 18.5 nanometers, which is roughly similar to the distance through the Z-disk. What does that actually mean? Because of course you see those plots. What does it actually mean? We have various different terms in order to be able to fit and create that plot. We have a background term. We have a localization precision term, because when you localize a molecule, of course, there is some error in that. So we have a localization precision term, and then these are the repeating distances that we think that we've modeled as you go through the Z-disk for where you would find a labeled alpha-actinin. And then that all sums up to give you that total model, which is then fitted to the histogram. And that pinky line you can see above the red line is an estimate of the error in that fit. So that's in X and Y. If you go into the YZ direction, it gets much harder. So it's much harder to see anything in the raw data. And Alistair spent a long time. So for this, he had to do a kind of smoothing to get anything out of the data at all, which is this kernel density estimation. And that's where he thinks he can pull out these 2 other dimensions, the 11 and the 24 I was showing you in the earlier diagram. But this is really difficult because, in fact, even though the Z-disk is supposed to be really nice and crystalline, it's not as crystalline as you would think, and it's not as ordered as you would think. And there are discontinuities in the lattice as you look in this direction, going through the Z-disk. So that doesn't help us. But we are able to get some sort of distances out. So that the real challenge will be to go on and look at some of the other Aphromoos that we're now developing to see whether or not we can see some different distances and different organizations, and really start to try and find out how all of the different proteins are organized. Because alpha-actinin is our kind of test bed. We know how alpha-actinin should be organized. So can we see differences or changes for other Z-disk proteins, and then do 2 color superres and try and put it all together. So that's what we're currently doing at the moment. So this is my last slide. It's really hope I showed you that Aphromoos are good for super resolution for all the various reasons that I've mentioned. And a few extra things from the Z-disk stuff, because we can do these co-crystals and find out very nicely where the Aphromoos are actually binding to our protein of interest. And what we're currently doing is trying to use Aphromoos to all the Z-disk proteins to try and find out how proteins are organized within the Z-disk to as high a resolution as we can using SuperRes. Well, thank you very much. I'm going to stop there, and hopefully I'm going to stop sharing. That's the easiest thing, isn't it?
Great, thank you very, very much. That was a great talk. I think Stephanie, are you ready with the quiz?
Yeah, as ready as I am always.
Ready as ever.
Let me just, I hope so.
So
I put the link in the chat, so I think you should see the link. So please sign up. Thank you, Michelle.
Oh no,
so
we've
are
got
you
to
now?
do the quiz now, haven't we?
Yeah, are you now a startup lady?
We'll do the quiz first because I don't want to answer one of those questions till we've done the quiz.
Okay, we do the. I hope I'm getting this. I'm always kind of fretting over this. I don't know why. Okay, share my screen. Oh, I can't share my screen. Oh. As I said, I have this new computer and it kind of makes me a bit. I can share a portion of my screen.
I can answer that last question about Aphromoos approaching A gold. Yes, you could absolutely do that, and that has been done. In fact, you just use the maleimide linkage like you would, but you, instead of putting a fluorophore on, you put a gold label on. In fact, one of the things we're hoping to do is to do that with the ZDIS and see if we can actually get some higher resolution from doing EM, and then that would also lead to the correlated LM-EM work.
are you getting anywhere with your screen sharing, Stephanie?
No, I don't know. It's kind of, let me just see. I need to go and do some stuff.
Right, well.
And the question about the RPD and the radial distribution function, I'm going to leave you to ask Alistair next week. There is I'm sure there is a, on the 1st of November, I'm sure there is a difference. That's really one for Alistair to answer. Yes, repositories for Affimer in the community. You know, this is I should say this is the one downside of Aphromoos. Aphromoos were invented at Leeds, and then the IP was generated at Leeds. And the unfortunate thing is that we would like to put all our Aphromoos onto AdGene, but the company that commercially screens for Aphromoos is happy for us to do that, but the university is not playing ball. They think they can make money out of the Aphromoos to Tubulin, which I doubt whether very much they were really going to make a lot of money out of it. So we keep trying to push this and trying to get the university to allow us to do it. But to be honest, probably what we ought to do is make a proper repository of all the Aphromoos we have, and others, perhaps Darren has, and you can just ask us. You have to sign an MTA, but you know, it's.
I wonder even if just you know, a repository to list, you know, the ones that exist.
Yeah, it's a good idea, isn't it? Because we've got quite a few now.
Of course, you know, as we work on KRAS, I just, you know, Google it and I found that there is one, but is you know, is always nice when you have a consolidated database curated, and nowadays the production of you know, the proteins is not really an issue. Is like an AdGene. AdGene is great not only to receive things that are already made, but sometimes also just to see what there is, and then you can synthesize. Yes, so it's if you want to modify it.
Yeah, I think we even had a talk with Janelia about them. They were happy to make them and distribute them, you know.
Yeah.
And for free, you know. And but the university just, it's hopeless because they just think, oh, if there's any slim chance they're going to. I mean, they're not going to make. The thing is, they're not going to make millions out of it. They might make a tiny bit of money out of it, but
well, they don't know because antibody technology wasn't really patented. No, that's always the biggest joke in Cambridge University or Cambridge academia. That I mean. Niels and Köhler were told that antibodies would never be a big deal and they would never make any money. So, I mean, who's to judge? I kind of opened my desktop and I'm sharing my screen so people can actually join now. If you could...
it's a very easy quiz.
Should we just quickly go through it? And my question was where you find the most exciting patterns as your first question. You can put in some breakfast, someone is saying, which in stock market. If I can continue with this. I don't know why it's giving me always such grief. This, and I had actually an idea for this Mentimeter quiz. I mean, how ridiculous. So yeah, there you go. Okay, so we have a few people only, but so I think just kind of if you want to just kind of comment maybe the kind of questions. Oh, it's in doing question 1. Why is it not showing question? Oh, okay. Yeah, it is. So this was a number question. So the there we go. So the question is what the size is of the linker. And you showed this was 20 nanometer, the linkage error for indirect labeling. Oh, God.
Well, it depends. It's a bit of a trick question because it depends exactly which method you use and how you label your antibodies. But anyway.
It's not okay.
We miss a little bit the music of that Curti had.
I know.
To fill
I'm
the
so
gaps.
sorry. Probably you can sing a little bit, so if you want. So these are 2 questions about what is not true, which is also a bit tricky. So you were asking what is not true. Are there small single domain proteins? Can they be expressed in E. coli? And can they contain cysteine residues? So people got that.
Of course, once you subclone them, they might very well have a cysteine residue in them, but that's the whole point, is that you can put it where you want it.
There's a second one like this, so it's not the same question. So there are 3 more questions here. So they have been used in expansion microscopy, they have been used in DNA paint, and no sharks or llamas are needed to make Aphromoos. Which one is not true? I think the people here have probably not quite read the question. This is an educational.
Well, you don't need sharks or llamas. So we haven't actually tested them in expansion microscopy, but I'm very interested to see if they work. And I think Helge Overs has got a nice new way of doing retention expansion microscopy. And I hope he's going to test our Aphromoos out in that form of microscopy. So that's actually trying to cross your Aphromoos into the gel. And something we want to try actually is, because you can biotinolate them, I found out you can buy streptavidin with acrylamide on the end. So actually that would be quite interesting to see if you could put the Aphromoos with the fluorophore. So you'd have to label on, put your fluorophore on the lysines, but then stick it into your samples, add the acrylamide, and it should co-polymerize in your expansion gel. This is something I need to get a project student in the lab to try out.
So this is another dimensional question. So what is actually the linkage error for the...
This is like a sales pitch.
Oh, fine.
Sales pitch for Aphromoos.
Yes.
If only it was easy to get hold of them.
I have one more question. Okay. So bear with me. So that's the last question. Maybe you can comment on this because I wasn't quite sure.
So again, I think again this is a bit of an advert for Alice's talk, because the Akaki information criterion was a really nice statistical approach actually suggested to us by somebody in statistics. It's used a lot in ecology apparently, because it's a very good way of comparing spatial distributions, because you're typically trying to model and you've got lots of parameters, and it penalizes you. The more parameters you put in, the more penalties you get. So it's a way of making sure, because of course you can get anything to fit anything if you put enough parameters in.
Okay, so we have one winner called Venture, which is maybe quite befitting, since kind of talking about kind of startups and entrepreneurial approaches to this. So I can stop sharing. Sorry.
Right. Okay. So Venture.
I think I probably answered most of the questions. The only other one was about the colour maps, and I think the colour maps, I don't know if that's the DNA paint one that Ralph made.
Oh,
in
yeah.
particular, which is really just to show, I think just to show the Z, really sort of highlight the differences in Z. And we sometimes use colour maps to show Z distribution as well. It's quite useful.
Well, that question was from Curti, and he's very interested in how single molecule localization data is represented in traditional images, and because there's a bit of a disconnect between the raw data and what we normally look at. So that was, I think that's what he was after. So I guess is the intensity in the color map giving you some? Is that indicating localization precision or
No, I think in that particular case, I think that was, if it's the one I think of, it was just indicating Z position.
Okay.
Yeah, but yeah, you could, I mean, that would be a good way to use color maps, wouldn't it, for precision, if you know that if you're looking at a lot of molecules and you know what the photon count is for each localization, and you know the precision is varying. I don't know if there's a straightforward way to do that though.
Actually, Curti, he's still there, because I know last time we saw Curti, he was in a taxi going through Calcutta. So I don't know if he can still hear us. No, okay. Well, we can move on.
So just the commercial supplier for Aphromoos, there is one. It's Avacta, but Avacta don't really supply them as such. I mean, I think they do sell some. So, for example, as you might imagine, they've made Aphromoos for COVID, for the virus that causes COVID, spike protein, etc. So they do work a lot with commercial companies, and they do have some Aphromoos on their site. But so there might be something there that you're interested in. So that's Avacta, that's AVACTA, but they don't do any of the ones that we've generated in the lab. And I think the other reason why they don't do that is because you really have to do the quality assurance on those. And yes, it's all right if I make some and tell you that it works, but maybe the next batch I make, there's something wrong with it or something. So you have to have somebody there to really kind of make sure that they're all tested and working before they send them out.
Okay, so did you do this question from Cynthia Vecui?
Yes, I said you have to ask Alistair that one.
All right, okay.
So we have a more detailed talk quite soon.
He's really good. That's really, I'm sure he'll have an answer to that. I'll probably ramble and make something up, but you might as well get it from the horse's mouth.
You had a question about expansion microscopy. Again, you've already had a mention.
Well, so as you can see, we haven't really tried that yet, but I really want to do it. I'm trying to persuade a project student who's still deciding what project she wants to do that this is the one for her. So fingers crossed. But as I said, and Helga Overs is going to do something hopefully as well.
All right. Okay. So actually, I have a question. So I think you mentioned live cell imaging.
Mm-hmm.
So what what do you think is going on there? I mean, presumably this is something you're adding sort of in the media around the cells.
Yeah, we just make a GFP expression construct. So we just take a standard GFP vector and put the Affimer in and make it as a protein and transfect that in. But in principle, I mean, so you can do that in other ways. So you can, there are all sorts of ways, because you've got the clone. For example, all the things we've done, we've made EOS fused to an Affimer and made that as a protein and used that in Superres. That works. If you don't want to, you know, so you've actually got it, you don't have to label on the cysteine, you've got EOS there if you want to do palm for some reason instead of storm. Actually, EOS and Alexa 647 does seem to work quite well as a combination, so that might be a reason why you want to do that. We have, you can put snap tags on if you don't want GFP, you can put snap tags on, halo tags, anything you can do with any protein, you can do it. But of course, with all of these things, if you make a peptide or something, or you're expressing something in a cell that binds to something else, there always is the danger that you'll interfere with the normal function of that protein, or you don't see the distribution of the protein that you would normally see. And that's certainly true of the GFP actin Affimers. They only seem to recognize a subset of the actin cytoskeleton. So, and you know, even LifeAct, which everybody uses, probably does have an effect on myosin binding in the cell to some extent.
Right.
And it does for some reason. Cells, when you express LifeAct in them, you immediately notice more phyllopodia. So there's something, you know, even LifeAct that everybody uses is not absolutely perfect.
So another one, there are some other sort of artificial antibody approaches as well. I'm thinking of things like bicycle peptides, for example. So is there any sort of merit or benefit or whatever? What's going on with these relative to Aphromoos?
I mean, there are some. So, for example, that we wrote a review about this stuff a couple of years ago, and at that point I was very interested in aptamers as well, which are really good for labeling DNA, RNA, but had also been used a little bit to label proteins. You can use those, and I think since we wrote that review there's been quite a few papers come out about their utility in labeling RNA and DNA, which I'm not so sure that you want to use an Affimer to try. I don't know. I don't think Darren's ever tried to make Affimers to DNA RNA. So if you wanted to label those, probably not much. So yeah, I mean, there are other scaffoldy type things around, but you've got to have a library, you've got to be able to screen it. You know, we just happen to have that screen facility set up really nicely at Leeds. So it's really easy for us. And, you know, anybody can use it. I mean, you just have to pay them a bit of money. But they're all screened, you know, they're quite happy for people to contact them with a screening project.
Okay.
I did a fast search. I didn't find much, but it could be interesting also to use them. I mean, I know that they're used as potential inhibitors, protein-based inhibitors, but also together with optogenetics, for example, to do a allosteric inhibition of the Affimer. I didn't see in a fast search that was done. That could be quite interesting.
Yeah, the other thing that we've had a little bit of a go at, but we still need to finish off, is an idea where you put a dye on your Affimer, and when it binds to your protein of interest, you can get it to transfer the dye onto your protein of interest using some clever chemistry. And we've got the first stages of that to work. But then, you know, it was one of those short projects where you don't have enough time to finish it off. So hopefully we will get that finishing off. But that would be a cool technique, because then you could also put a tag on your Affimer so you could express it in the cell, get it to transfer the dye, and then degrade your Affimer, and then you're left with protein that's in a fantasy world. That's what we'd like to do.
Right. Just a quick one. What was the molecular weight of these things?
They're about, well, it depends exactly what the sequence is in the loops.
Right.
They're roughly about 10 to 12 kilodaltons.
Okay.
Pretty.
So they are pretty small. Yeah.
They are. Yeah.
All right. Do we have any other questions coming in? I think we've been through everything in the chat so far. If anyone wants to put their hand up.
And if anybody wants to try the actin Affimer or the Tubulin Affimer out, just write to me and we can organise getting some sent to you.
This could be the prize for the quiz winner.
Oh, yes.
A little tube of Affimer instead of a Foldscope. I mean, please specify when you won, when you won the quiz. Maybe we can offer.
You have to sign an MTA first. That's the only
yeah,
thing.
that's fine.
But it's
You can get the Foldscope without an MTA.
yeah.
Well, we're almost family here, so I guess.
Okay.
Thank you very much again, Michelle. That was wonderful.
No problem.